This is part 3 of the video on angiosperms, work smarter, not harder. In this one, we'll look at different types of inflorescences. You'll learn more of these in lab and learn how to identify them better, and then we'll look at the angiosperm life cycle. I should say, and I'll say this again later, I'm not going to cover all the different types of inflorescences. I'll cover some common ones, but much like the angiosperms being a diverse and wonderful group, there are many different types of inflorescences, and many of them are sort of compound and confounded with each other. So almost every option that exists, you'll see in the angiosperms because they're trying it all out. So when you think about an inflorescence, you're thinking multiple florets where a single flower would normally be. So if you have a peduncle, and then that peduncle doesn't end with just a single flower, a beautiful flower, but instead you get many flowers coming where that would be, and you can sort of identify that again based on nodes, because this single flower might have leaves that led up to it. Maybe it would even have branched flowers, but each of those would emerge from a node. So what you're looking for are nodes, because flowers are going to emerge from that bud position, whereas if we have a leaf here or a leaf scar, and then a bunch of little flower-like structures that emerge above it, but none of them has a node, then we know we have an inflorescence. So be looking for nodes to identify whether you have a single flower or whether you're looking at an inflorescence. So in an inflorescence, each of these is called a floret. And now, instead of having just the peduncle, which still exists, we'll also have these little stalks that lead up to the florets, so they need their own name, and that's the pedicel. So we have florets with pedicels, and we have flowers with peduncles. You can think of an inflorescence as analogous to a compound leaf, whereas with a simple flower, we're looking for, it comes from a node, right? The branch or the bud location in a node, whereas with an inflorescence, that will also stem from that bud component of the node, but we won't have other nodes within the inflorescence. Same for with compound leaves, where we had simple leaves that came from a node and from the leaf location, whereas when we had compound leaves, it came from the node in the leaf location, but it didn't have its own buds within it. So that's kind of the same thing you'd be looking for. This first type of inflorescence that we'll look at actually just looks like a simple flower, and you have to look pretty close to see that it's not. So there's a big receptacle that's at the top of the peduncle, just like normal, and then we have structures that look like sepals on the outside, but they're not, because this isn't one flower, it's many florets all attached to the same receptacle. And that's going to be called a head inflorescence. You might also see it called a capitulum. Oh gosh, I'm going to spell this wrong. Capitulum? No, it's only one L. There we go. Okay, so it's a head or a capitulum, and that's many florets that are all attached to that same receptacle. So it's at the top of a peduncle, and then these structures that look like sepals are called filaries. So each one is called a filary, and together they make up something called an involucre. These are super common flower types, and now you're going to see them everywhere. Dandelions are a head inflorescence. Daisies are a head inflorescence. Everything that looks like it's related to those is a head inflorescence. Thistles are a head inflorescence, and there can be all different types. So a dandelion is composed all of these longer florets. So these are asymmetrical, they're zygomorphic. So we would have a single strap-like flower, and on a dandelion it would have 5 lobes, I believe. It's going to be a ligule, and then it will have the other flower components that come out of that. But this is your corolla. and it's all fused together. And you can count how many fused petals there are by how many little nodes you have on the top or the end of that thing that looks like a single petal. So it's this fused corolla, and that would be a ligulate head where it has all of these five fused petal flowers, or florets, sorry. So a dandelion is a ligulate head. Whereas if we look at this picture we have here, I'm not sure if this is a sunflower or just a daisy, but we have these little florets in the middle that are very different from these big florets on the edges. Those little florets in the middle are called disc florets, and they have radial symmetry, where if we're looking at it from the top, we can see that there's 5 petals. I'm not going to be able to draw something radially symmetrical probably. And then our little floral parts would come from within that, right? So we have these 5 petals that do have some fusion because they make a little bit of a tube, but they're much easier to count. So we have disc florets in the middle, and then on the edges we would have florets called ray florets, and those are typically divided or composed of 3 fused petals. And they are also bilaterally symmetrical. So head inflorescences are complex, but worth learning about, because if you ever go into plant ID, this will be many of the inflorescences that you find. Another type of inflorescence that's pretty common are umbels. So an umbel, I always think of this like umbrella. It's kind of like if an umbrella gets blown inside out. We have our receptacle here, and then all of our pedicels attach at the same point to our receptacle. So that pedicel attachment to that single point at the top of the peduncle or receptacle, that is an umbel. You might also see compound umbels, where you have all of these pedicels that lead to a bunch of other little kind of compound florets coming from their own little umbel. So you can have an umbel, like over here, or you can have a compound umbel, which is umbels at the end of all of your pedicels that come from an umbel inflorescence. So many, many ways this can go. In a spike inflorescence, all of your florets are attached. So we have our peduncle here, and then we just start to see florets attached all the way around it, and they're directly attached to it. So each of these florets would just end on this sort of peduncle structure. This whole thing would just be like one big receptacle, basically, and all of the florets are directly attached to that. We don't have any kind of stalk that's coming from that receptacle structure to attach those florets. That's a spike. That is different, but very similar to what you're seeing over here on the right, where we have a stalk that attaches the floret to that receptacle. So that, where we have this kind of spike with pedicels, we call that a raceme. And you can also have branched racemes. Those are called panicles, and we'll see spikes within other inflorescence types. So spike, no pedicels, raceme, pedicels. So here's an example of where you see a spike type structure within this inflorescence that'll have a different name. So this is a spathe and spadix. This you can see pretty commonly with skunk cabbage or with calla lilies. So this thing that you would think of normally as the corolla or the kind of showy flower structure, this is a big leaf really that subtends our inflorescence. So a leaf that subtends something is called a bract. So we have this showy bract that makes the sort of floral appeal, and then all of our florets are actually within this structure. So this is a spathe, and I remember that word because it sounds very round, spathe, and I think of that just when I kind of twist my hand around as I say it, spathe, because it has this sort of wraparound feel to that word. So the spathe is this showy bract that wraps around it. It's not actually part of the inflorescence. The inflorescence would be down here, where you can see we have pollen-producing florets, and then we have these pollen-receiving florets, and I should use a different color here. So these are the female florets, and you can tell because they have little knob-like structures on top of a rounded structure. So we can think of our little cartoon of what that looks like, right, with our ovary and our stigma. Up here, we have these kind of elongated sporangia at the end of a stalk-like structure. So these have stamens, where we have anthers on the end of our filaments. So each of these florets is a unisex floret, and in floral terminology, we would call that an imperfect flower, and it's incomplete. So any flower that is missing a whorl is considered incomplete, and if it has only one of the sexed whorls, so if it only has a gynoecium or only has an androecium, we call it imperfect. So a spadix is basically a spike inflorescence of these imperfect florets. So as you can see, those are just some of the inflorescences that exist out there. There are many other types of inflorescences that you might see that are similar to these. It's like a panicle is a branched raceme. Cymes have to do with your side growths produce your new flowers or florets. So every time you kind of have your terminal flower or floret, a side growth is going to produce another floret, and so on and so forth. For a corm, you have these flat-topped inflorescences, and then I actually don't know what a thyrse is. Maybe it has to do with having a compound helicoid cyme. I don't know. What's a thyrse? I'll have to look it up. Catkins you'll see a lot. This is maybe the most common one that we didn't talk about, where you have a bunch of little florets that it's basically like a spike out on its own, except for it'll be unisex. So you'll have male and female catkins, and these are common on a lot of our wind-pollinated trees like birch and alder and willow. The angiosperm life cycle is complicated. Of all the life cycles we've learned, it's definitely the most complex with many different parts. So we can't really draw it in that same meiosis, fertilization, kind of haploid, diploid way that we do, because there's even a triploid stage. So I'm just going to draw the individual kind of components of our gametophytes or gametophyte formation. And we'll start with the megagametophyte. So if you think about floral anatomy, I'll just draw a little aside up here. We had a pistil, or our fused carpels here, with the stigma at the top, right there, on top of the style, and that sits on top of the ovary. What we're going to look at is a cross-section through this ovary. So here's our ovary, and let's say that it's 3 fused together, and we'll have these 3 locules. So in one of these locules, I'll draw a couple of our forming ovules. Okay, so this is all ovary tissue here. This is our ovary wall. This is a locule, which is just a compartment within a fused ovary. And then within this structure here is our megasporangium. So we have, I'll draw the integument here in red. This is kind of protective coating. We have the funiculus here in orange. And this attaches the developing ovule to the ovary wall. And where it attaches to that ovary wall is this sort of transitional site where nutrients can be kind of passed from the sporophyte to the developing gametophyte. And so that, much like in humans, is called the placenta. So those are three components that we have going on. And then we also have the green, which is our megasporangium. So we're going to look inside that megasporangium. Here's our developing ovule. So the megasporangium is there in green, and inside that we would have a diploid cell that would be the megasporocyte or the megaspore mother cell, called the MMC. So the megaspore mother cell, which is diploid, or the megasporocyte, will divide by meiosis. So that's where meiosis happens, just like usual, inside the megasporangium, and it'll happen inside the microsporangia as well. So meiosis happens there, and that is going to produce four cells that are haploid and all genetically unique. 3 of those cells, just like before, are going to die. So now we have this megaspore, which is haploid, and these 3 die. And just like with our gymnosperms, this opening here is called the micropyle. And that is where the pollen tube is going to enter the megagametophyte and fertilize it. So a complicated set of interactions is about to happen. So this megaspore is going to go through a few rounds of mitosis without doing cytokinesis for all of those. So if we look at our sort of transitional states, we will end up with 8 nuclei. So I'm going to draw these 8 haploid nuclei in here. Within this kind of large sac. And those are going to wall themselves off and become individual cells. And this is still our micropylar end down here. So we'll have the egg cell, and that will be flanked by 2 synergids. They help guide the pollen tube to the egg. I think the pollen tube actually enters one of the synergids, and then that synergid explodes, and then that triggers the pollen tube to stop growing, and then the sperm get deposited. So it's complex, and I don't actually know if anybody knows the function of these 3 cells back here. These are called antipodals. And as far as I know, they don't do anything. They might be kind of like our prothalial cells where they're vestigial, but they do get carried on into the mature female gametophyte. So they don't disappear, but I'm not sure if currently we know what their function is. Then in the center of this, there's a large central cell, and this is the weird one because it has 2 nuclei. I'm not sure if you'd refer to it as dikaryotic. We generally refer to it as just diploid. I don't know if these 2 nuclei have fused with each other or not, but we always draw them as separate. So I would think that it's dikaryotic. This is a central cell. Oh yeah, they're definitely separate because we call these the polar nuclei. So all of these cells are haploid except for this central cell. So when a pollen grain, we'll draw that over here. So this is our pollen, which is the microgametophyte. And now it only has 2 cells. So the pollen will have, I'm trying to use different colors, but I'm running out of colors. Pollen will have a generative cell, just like before. And then the rest of it is the tube cell and that tube cell nucleus. So we only have 2 cells. And this pollen is going to land on the stigma. So if we draw over here, our carpel like before, the pollen grain would land on the stigma, and then it would grow the pollen tube all the way down into the ovary, which would then have to find its way in through the micropyle, and it's being directed by the synergids, and I think the central cell has something to do with it too. So our pollen tube grows in here, hits one of these synergids, explodes, and then the 2 sperm cells that were produced by this generative cell, which just divides by mitosis because it's already haploid. And then these 2 sperm cells are each going to fertilize something this time. One of these sperm cells is going to fertilize the egg, while the other one is going to fertilize the central cell. So what we'll end up with so this is after fertilization. We'll still have our antipodals. Our central cell will now have a single triploid nucleus, and it's called the endosperm, this large cell. And then we'll have a fertilized egg, which is now called the zygote and is diploid. And I believe both of the synergids get burst. However, I don't actually remember. So this is our kind of important stage, and now it's a little bit chaotic because I drew the pollen in there. But this stage here, where you have 7 cells with 8 nuclei, this is called the embryo sac. And I'm going to link a really great walkthrough of the angiosperm life cycle because you really need like 20 minutes to cover this. This is what we're talking about, right? We're talking about fruits and fruit development. So here we have seeds. This is what would have formed from that ovule. The coating on that seed would form from the integument, and then within that we would have our developing embryo. And you can see that these seeds, which form from the ovule, still have the funiculus and this spot where they're attached, which is the placenta. So the ovule gets fertilized, it becomes a seed, and then this whole structure, that ovary, is going to develop into this fruit. So now we have a fruit, which this would have started out as a large flower, but still, you know, a flower you could hold in your hand. And the ovary swells up gigantically, which we've also selected for, to then get animal dispersal for these seeds. And I like this picture because you can see different locules. So we have 3 different locules here, and they just have a really clear funiculus. Here's an overview of the angiosperm life cycle from your book. You can use this or not, but we will cover it more in lab.